Welcome to Corning Cells on the Mesa presentation for the Alliance for Regenerative Medicine virtual event. My name is Keith Olson, and I will be introducing Corning today and some of our enabling technologies for cell and gene therapy. I hope that many of you are familiar with Corning and our life sciences products, and I'm sure that you do know us for some of the glass that you have on your favorite devices. Corning is a multinational Fortune 500 company with approximately $12 billion in revenue last year that serves multiple industries around the world. Our strengths are in manufacturing, material science, technology, and innovation. The life sciences division comprises about 10% of the overall corporation, and we have a rich history of innovation dating back to the early 1900s. Corning is no stranger to challenges or emerging technologies and the demands they present. And our products have helped drive life-saving innovations from the production of penicillin and the polio vaccine to the advent of cell culture and growing cells on plastic, and now newer advances in the life sciences industry. So how are we achieving this? We have built our laboratory consumables business on a foundation of highly recognized brands and a reputation for quality. During this time, we have established strong core capabilities and expertise in cell culture and bioproduction. Those capabilities are now poised to help Corning work with our customers to address the challenges ahead for new cell and gene therapies. Today's presentation is going to focus on two specific areas: the production of viral vectors to support cell and gene therapies, and a significant workflow advancement in cell isolation and processing. Both of these areas present some very interesting challenges, and I will now walk you through the solutions that are going to be available from Corning. So let's start by talking about the Corning Hyperstack. This was a technology we actually launched quite a number of years ago that was developed to provide researchers with a compact device that was able to produce billions of adherent cells in a closed system environment. It introduced a step change in cell expansion compared to the alternate 2D vessels that Corning and others manufactured. And what we did was significantly improve the space utilization for cell growth by up to 3-fold in many examples. Turn the clock forward a few years, and we found ourselves with a product that was the ideal solution for producing AAV and lentiviral vectors at a time of surging demand needed to support the emerging new therapies and the successes that they were having in the clinic. Adherent cells do have some advantages when it comes to transfection and transduction compared to cells grown in suspension. And the Hyperstack presented a simple system with low capital demands to start producing large amounts of virus effectively and efficiently at a modest scale. So it found a lot of popularity for this emerging application. Each Hyperstack 36 unit is capable of producing on the order of 10 to the 14th viral particles, depending on the vector and the process. So it makes it a very efficient starting system for these applications. As you can see in a case study example from WuXi, this product has been successfully used in cGMP production environment to produce large quantities of viral vector to be used for gene therapies and gene-modified cell therapies. While the products are simple to use and multiple units can be processed together to make a batch, this type of technology is a scale-out approach, and eventually you meet a limitation based on overall production demands. The cell stack image in the second slide illustrates this challenge, I think, very well, showing you the scope and scale of what is required to mass produce virus using 2D stacked vessels. This presents a challenge overall for the viral vector production market and has driven the industry to work very hard to adapt this process to more scalable platforms. Corning also recognizes the scalability challenge, So we are launching a new platform to address this need while incorporating all of the benefits that are inherent to adherent cell culture. The new platform is called the Corning Ascent Fixed Bed Bioreactor. It has been designed specifically with viral vector production in mind, and with the eventual capability and capacity to produce 10 to the 18th viral particles per batch. The image shown here represents the process development scale unit, but the intention will be to use the same technology and process to scale this new platform up to an internal surface area of at least 500 square meters. While there are other fixed bed bioreactors available and in use today for commercial production, users are still facing challenges with these platforms. Benchmarking has confirmed that our process development unit has outperformed the leading competitor in a number of areas, and this is driven by the key improvements we have made to the workflow, cell harvest, cell uniformity, and the actual cell growth within the fixed bed itself. And together, this has yielded better results in viral yield, a reduction in the absolute number of vessels to produce virus, time savings in go-to-market, and a significant reduction in the amount of media used to produce final product. So we're very excited about this new approach and what it has the potential to deliver. This slide is a summary, now presenting when we will launch this new technology. The process development scale unit, which has the ability to scale up to 5 square meters of surface area, will launch in the first quarter of 2021. In addition to the performance benefits I've already highlighted, the product is also going to present significant annual cost savings and truly provide an adherent cell culture bioreactor that can perform at the production scale. With the remaining time, I want to transition now from the viral vector landscape to a discussion on cell isolation and a new product manufactured by Thermogenesis that Corning has partnered on for commercialization. The XSeries products are a range of solutions designed to automate, simplify, and improve current cell separation methods. They not only make the procedure safer and more effective for use in workflows like cell and gene therapy, but they are also much faster and reduce labor costs. By adding the novel capabilities of the XBAX technology, which I'll describe further in another couple slides, cell separation can now be taken to the next level of cell isolation to identify and select specific target cell types. Existing workflows for cell isolation and cell separation are met with many challenges, including variability, labor intensity, safety, and compliance with good manufacturing processes for cellular products. The XSeries addresses a number of these concerns with their novel technology, and all of this operates on a simple benchtop swinging bucket centrifuge. The best way to think about the XSeries unit is as a smart centrifuge bottle. The vessel itself is a multi-chambered unit that uses simple centrifugation to separate cells based on density and segment cells into 3 separate chambers in the unit, which you can see here. In the example shown for whole blood, at the end of the 35-minute process, you are left with red blood cells isolated in a depletion chamber on the left, the target mononuclear cells isolated to a collection chamber on the right, and the remaining plasma and other blood components in the central sample chamber where you loaded your original sample. All of this separation is achieved with the reusable control unit at the base of the chamber that provides the smart features necessary for this process. Comparing the data to the predicate method of Ficol gradient separation, you can immediately see the improvements delivered in consistency and recovery while separating MNCs from whole blood. Furthermore, you can process up to 4 samples simultaneously in the same centrifuge using the X-Series products, allowing the approach to deliver significant time and cost savings in addition to the inherent benefits of doing all of this work in a closed system with minimal human interventions. The entire previous example was for whole blood and required no added reagents, just the unit. Separation was achieved naturally inside the unit using the cell's differential density. However, this technology has the capability and the capacity to be extended with an innovation in cell isolation using microbubble technology. This is called buoyancy activated cell sorting, or BACS. Using standard antibodies for cell surface markers and traditional biotin streptavidin chemistry, BACS cell isolation is achieved by binding microbubbles to the target cells of interest and having those specific cells float in the central chamber until you're ready to run the collection procedure. After using a very similar protocol to what I just showed you for whole blood, all the non-bound cells are separated in the depletion chamber. The microbubbles are then subsequently popped gently and non-invasively, not harming the cells, to release the target cells, and they are then isolated to the collection chamber, as you saw in the previous slide. In closing, the X-Series portfolio can introduce significant changes to the standard approach for separating, washing, and isolating cells from complex solutions such as whole blood or leukapheresis. It comes with the added benefit of onboard record keeping and minimizes the amount of human variability that you see in all the other approaches. So I want to end by first saying thank you to the organizers of this virtual event, which I hope is very impactful and is able to help all of us who can't travel at this time. And I want to thank all of those who are watching this for taking their time and giving their attention to Corning to see our latest innovations. I encourage you to please visit our website or feel free to reach out to me directly with any questions or other inquiries using the links provided here. Thank you very much, and good luck with the rest of the conference.